Okay. So good afternoon to you all. So I welcome you all for the technical session on food, nutrition, preservation, value addition and post-harvest technology. So I'm Professor Devahi Mahendran. I'm the chairperson of this session and I welcome my panel members Dr. Champati Gunatilaka and Dr. Mrs. Bavadarani. So we are a little bit behind the schedule as because that inaugural session was little bit delayed. So shall we start our first paper? The title of the
Recording in
paper is
progress.
Development of Pickles from Yellowfin Tuna Mussels and Changes of Nutritional Content During Storage. And the presenter is S.M.S. Afrin. Hello Afrin. Afrin. Unmute your mic system. Okay. Okay, Afrin, since you get ready, I think we will move on to the next presentation. Vidana Pathirana, Miss, are you ready? Miss Ansala?
Actually, I am the one who is behalf of the Dr.
Okay, no problem. Okay, so you can present effect of leaf maturity on the antioxidant activity of Moringa oleifera in the dry zone of Sri Lanka. Okay, now you can present your research findings. Okay, you can start now.
That means the video does not play, right, madam?
No, video is not played. Can you share? Do you have slides?
Okay.
Okay, you can share your slides. Okay, we can see the slides. Thank you.
Okay, can you hear me clearly? I hope.
Yeah, we can. Yeah.
Right. Okay. Thank you very much. Good afternoon, all of you. I'm Supun Bansekara, and I'm here to present about my research findings, current research finding, which is the effect of leaf maturity stage on the antioxidant activity of Moringa leaves, especially in the dry zone in Sri Lanka. So before moving to the technical details, I would like to give kind of a brief introduction about the antioxidant and the sector. So the antioxidant is provide protection against normally non-communicable diseases, which include cancer, coronary heart diseases, kind of diseases. So previously, synthetic antioxidant like BHA and BHT have been used for a period of time over the years. But recent report have shown that the safety concern about the usage of those kind of synthetic antioxidants. So as a result of that, natural product that have gained worldwide attention as a antioxidant product. So likewise, many herbs, fruits, vegetables, spices, and legumes have been exploited as source of antioxidants. Similarly, Moringa also have gained extra attention regarding the antioxidant properties, and recent research have been published based on the antioxidant activity properties of Moringa. So when considering about current situation in Sri Lanka, also there are several kind of value-added product has been developed through Moringa products. But the thing is that still we do not touch that commercial level production. All the production have been conducted in a small scale level. So the present study is mainly focused on to develop large scale production, and what is the correct maturity stage that can be used to obtain maximum utilization of that antioxidant properties. That is the purpose of the study. So the present study was evaluate the antioxidant activity of Moringa leaves under 3 different maturity stage. So the objective of this study is to evaluate and compare the antioxidant activities of Moringa leaves under 3 different leaf maturity stages. Those were tender leaves, premature leaves, and overmature leaves. So the experiment was conducted basically in 3 laboratories. One is CIARS laboratory for the extraction procedure, and National Cinema Research and Training Center for the food extraction preparation, and Agriculture Biotechnology Center for the antioxidant activity test. The samples were mainly collected in dry zone area, including Semanagala, Hambantota, and these areas. And leaf samples were collected in 3 maturity stages, and 3 biological replicates were used in the study. So the study was mainly conducted by using food extract from the leaves. First, all the leaves were collected, and fresh leaves were collected, dried, and grounded into small particles. Then extraction was conducted by using methanol, and 24 hours was extracted as continued under the 60 Celsius temperature. Then after that, the methanol was evaporated by using rotary evaporator, and then food was extracted. So antioxidant activity of Moringa leaves basically expressed in a percentage of DPPH discoloration method and IC50 value. For the purpose, we developed different kind of extraction series by using Moringa crude extraction as 20, 50, and 100 micrograms per milliliters. For the study, we used gallic acid as a reference sample to compare the antioxidant activity of different kind of leaf maturity stages. So antioxidant activity was the discoloration was evaluated by using UV visible spectrophotometer under the UV length 517, and the antioxidant activity were expressed as 2 methods, as an inhibition percentage, and as a IC50 values. So these are some figures that I have collected different maturity stage of leaves, and Moringa leaf powder and Moringa leaf crude that I have extracted. So basically the experiment was conducted as by using 3 biological replicates, and as well as 3 technical replicates also used for the study to reduce the error. And the data analysis, the antioxidant activity inhibition percentage were analysis using descriptive statistical method, and IC50 values were fitted into ANOVA table, and mean separation was performed according to the Nanat method. So if we move into the result, this figure shows the DPPH inhibition percentage values under different leaf maturity stages based on 3 different concentration values. Concentration values are 20, 50, and 100. The thing is, we need to grab in here is that with the increasement of the concentration of each and every maturity stage, the inhibition percentage also increase with the increasing of the concentration value. Further, it shows that highest inhibition percentage was shown by the gallic acid, that means standard, and then mature and overmature leaves. Here, this figure shows the IC50 values of the different maturity stage of moringa leaves. And according to that, IC50 value means the lowest value means the highest antioxidant activity. So according to that, again, gallic acid, that means standard solution shows the highest antioxidant activity. And here, interestingly, both mature and over-mature leaves does not have any kind of significant difference. And the lowest antioxidant activity or highest IC50 value was observed from tender leaves. So that means the highest antioxidant activity can be observed from the over-mature leaves and mature leaves compared to the tender leaves. So if we compare these values with the other species that are commonly available antioxidant properties, which are extracted by using the same method that I have used, that means methanol crude extract. So according to the data, we can see that moringa leaves, especially mature leaves, has considerable amount of antioxidant activity compared to ginger and anora. And as well as compared to gallic acid, also it contains considerable amount of antioxidant activity, especially in the methanol extract. So if we brief about the results, the highest antioxidant activity shown by the gallic acid and followed by the mature leaves and lowest by the tender leaves. And there is no significant difference between mature leaves and the over mature leaves. And as well as the study shows that the antioxidant ability increase with the maturity, and the reason could be the responsible biochemical compound for antioxidant activity increase with the maturity. And if I conclude all these findings into one slide, there was considerable and compatible antioxidant activity in moringa leaf compared to the gallic acid, which is the reference sample. And then the antioxidant activity of moringa leaves change with the maturity stage, and highest value shown at the mature and over mature stage. That means we can, if we are going to a kind of a commercial production or any kind of things, we can mainly focus on mature and over mature leaves for the better antioxidant properties. Further, the current study was conducted as a preliminary one. So based on the study, I would like to give some few suggestions for the study as well. So it is recommended to study antioxidant activity and as well as total phenolic content of moringa in different environmental conditions, especially under the different agroecological zones, what happened to antioxidant activities and total phenolic content. And also it recommended to study the different plant parts for antioxidant activity, especially for the nodes and all other things. So these are the references that I have used, and I would like to thank for listening my presentation, and thank you.
Okay, thank you for your presentation on the antioxidant activity of different maturity stages of moringa leaves. And there were a lot of research was conducted on the moringa leaf as value addition on iron enrichment, and some of the antioxidant activity was carried out in different plants part of different plants, like as what you said in the reference, anona and other plant part, nelly, gooseberry and other plant product. Okay, now this topic is open for discussion or any clarification. You can ask the presenter. And our panel member, Dr. Champadi, Gunatilaka, if you want to say anything on this presentation.
Yes.
Yeah.
Yes. In antioxidant, that it is clear with this presentation. So
Yeah.
I just like to ask that what is the temperature that antioxidant is getting deteriorated while that anything is processing that it requires some thermal treatment. So what is the maximum temperature it can be tolerated? That without
yes, according to the sir, can you hear me?
Yes, I can hear you.
Yeah.
According to the literature, it's mentioned that nearly 80 is the Celsius that it can be deteriorate. So in the study, I use as 60. But actually, if we use for hot water, actually when we are preparing something, for example, as a tea or something like that, definitely we need to use water. So methanol is not a solvent that can be used. So when we use water for a 60 Celsius, it need to be, kept for a while. So that is the thing I have observed. According to the literature, it says that 80 is the maximum level regarding the properties of antioxidants. So in this activity, I use 60,
Okay.
the temperature.
Okay. Yes. Thank you for your clarification.
So up to 80 is stable, huh?
It's
The
stable.
stable.
Yes,
Yeah.
up to 80
Okay.
is stable.
And I have one question, that how did you differentiate the selecting of samples, like, okay, premature mean very young leaves, mature and over mature?
If there is a one or two, leaves which is
Yellow.
increasing to yellow in color, not
So
all
that-
the leaves were getting into yellow color, but 2 to 3 leaves were getting into yellow color. I take it as a over mature leaves.
Over mature and other, oh, okay. Thank you, okay.
Okay, thank you very much.
Can you hear me? I just have one clarification to ask you. Uh, in your recommendation, you have suggested to check different parts of the plant for the antioxidant concentrations. And how about the varietal difference? Did you check different varieties?
No, actually, that is a huge gap regarding moringa industry. Currently, we have no any germplasm also. So actually,
Uh-huh.
we have to collect some data. Different kind of morphotypes are available, and based on that also, I hope that antioxidant activity and especially the total phenolic content, all those might be changed. So it is very important area to study the different kind of morphotypes and all these things.
Yeah, it may be a broad study and it may
Yes,
consume a
definitely.
lot of time. Yeah, it's a very good initiative study and it's very informative.
Yeah,
I would like to congratulate you on your good work.
Yeah, varietal difference, because there are some short fruit variety
yes.
and very long nowadays, the genetic modification, right? Yes, you can try, but the germplasm purity is the question. Yeah, as what you
Yeah,
said.
that is the.
Yeah.
I actually I observed. I collect data from different research institute, but
Yeah.
actually I cannot found such a germplasm.
Yeah. And sometimes our murunga 3 feet or more than that, sometimes very
Yeah,
large.
definitely. There are different kind of morphotype based on the leaf shape. Also, there are different kind of morphotypes and fruit shape and
Yeah.
branching habit. We can visually observe those things, but actually, it is very important to study in detail those areas. Even if we are going to, produce some commercial products and to develop this industry in future.
Okay, any more questions from the audience? Okay, since there's no question, thank you for your presentation on the antioxidant activity of different maturity stages of Moringa oleifera. Okay, so we shall move on to the next presenter.
Okay, thank you.
Okay, thank you. Identification of acetic acid bacteria from naturally fermented banana vinegar.
Yes, madam.
Harichandra, yeah,
Can you hear me?
yeah.
Uh, yeah. I will share the presentation.
Yeah, please.
Can you see the presentation now?
Yeah, we can, yeah.
Good afternoon, everyone. I'm Udeshika Mihirangi Harishchandra from Department of Food Science and Technology, Faculty of Agriculture, University of Rohuna. I'm here to present about identification of acetic acid bacteria from naturally fermented banana vinegar. So this is the content that I'm going to present today. Now I will go to a brief introduction about acetic acid bacteria. Acetic acid bacteria are gram-negative bacteria. and they are considered as aerobics or obligatory aerobics. They can be commonly found in products such as vinegar, fruits, and also in rotten fruits. They have the ability to oxidize sugars and alcohol to organic acids, being their final products such as vinegar and kombucha. Presently, the acetic acid bacteria can be classified into 10 genera. In natural fermentation of vinegar, the starch culture of mother vinegar will not be added, and the fermentation will take place as a result of acetic acid bacteria, mixture of acetic acid bacteria strains or single strain in the atmosphere. When it comes to the problem statement of the study, natural fermentation can occur due to different acetic acid bacteria species. The species of acetic acid bacteria responsible for fermentation may change according to the geographical location also. Thus, it is very important to identify the responsible acetic acid bacteria in natural fermentation. These are the objectives of my study. The general objective was to isolation and identification of acetic acid bacteria from naturally fermented banana vinegar. And the specific objectives are to isolate acetic acid bacteria responsible for natural fermentation of banana vinegar, to identify acetic acid bacteria through biochemical properties. This is the methodology that I followed. This study was conducted at the laboratory of Department of Food Science and Technology, Faculty of Agriculture, University of Rohana. So first I isolated acetic acid bacteria using YPG medium and YPS medium, because normally there are mainly two types as Acetobacter and Gluconobacter that can be present in vinegars. So to isolate those two bacteria, I used YPG medium for Acetobacter and YPS medium for Gluconobacter. These are the cultured bacteria strains. For the biochemical and physicochemical methods of identification and characterization, I use gram straining, catalase test, acetate and lactate oxidation, acid production from ethanol, carbon source utilization, acid production from various sugar sources, production of dihydroxyacetone, overoxidation of ethanol, growth at 30, 37 and 40 degrees Celsius. When it comes to the results of my study, I could isolate four strains. All the 4 strains gave gram negative for gram straining, catalase positive for catalase production test, acid production from ethanol was also positive for all the strains, and acetate oxidation gave positive while the lactate oxidation gave negative result. And all the strains were negative for dihydroxyacetone production, and all the strains gave good growth at 30 and 37 degrees Celsius respectively. When considering this biochemical and physicochemical identification test, the genus of the isolated bacteria was identified as Acetobacter. All the strains could produce acid only from D-glucose and ethanol. From the other sugar sources, it gave negative results. The carbon source utilization pattern also could be identified only at glucose. So, when considering the carbon source utilization pattern and acid production from sugar sources, it was identified that the isolated bacteria responsible for this natural fermentation of banana vinegar was Acetobacter pasteurinus. This slide shows the species strain level species identification. All the 4 strains were identified as Acetobacter pasteurinus. The final conclusion of this study was the isolated bacteria were identified as Acetobacter pasteurinus. These are the references which I referred in when conducting this study. Thank you for being an attentive audience. Now the presentation is open for discussion.
Thank you, Harichandra, right, for your presentation on the identification of acetic acid bacteria from naturally fermented banana vinegar. Okay, now it's open for discussion. Any questions or any clarifications from the participants, audience? So, can you go to slide number 9? Harichandra, slide
Okay.
9.
Is
Yeah,
it-
there's a small correction on your table 1. Calcium carbonate.
Yeah. Sorry for that mistake.
It's okay, it's okay. Right. And then you have tried different temperatures, no?
Yeah, I have tried at 30, 37 and 40 degrees Celsius.
So 40 degree, there's no bacteria. I see. So what is the highest tolerant temperature for Acetobacter?
It's something around 37 and 36.
Around 37, 36, yeah. So whether have you prepared the vinegar?
Yeah.
The methodology and all you have not shown, no?
For this symposium, I have only written about the acetic acid bacteria isolation part for the extended abstract. That's why I didn't include them in this presentation.
So this is a part of the research on the banana vinegar, is it?
Yeah. Yeah.
Okay, thank you. Any other clarifications? Okay, thank you. Since there's no questions, right? Thank you,
Hello.
Harichandra.
Hello. Can I ask a question?
Yeah, sure. Yeah.
I want to know when can I get the proceeding of the published extended abstract?
Yeah, today we have released.
Okay.
Hopefully we will upload today or tomorrow.
Are they sent to our? Will they
Now,
send it?
soft copy you can check our website.
Okay. From the website, we have to download the soft copy, no?
Yeah.
You won't send it to our email addresses?
Then anyway, I will check with the coordinator and the secretary. They will inform you.
Okay.
I will convey the message.
Okay.
Today we have already released in the inaugural session.
Okay. Thank you.
Okay, they will inform you. Yeah, sure. Thank you, Ms. Harichandra.
Thank you, madam.
Okay. Okay, we will move on to the next presentation. Afrin, please wait. You can present it at last. Okay, the next presentation is by Adhikaram. The title of the presentation is Determinant and Impact of New Technology Adoption for Pepper Post-Harvest Processing on Farmers' Well-Being, a case of pepper farmers in Kekol district. Okay, you can present your research finding. Is there anyone going to present on the topic that I have already mentioned? Okay. Okay, then we'll move to the topic number 1. Afrin, are you with us? Okay, we'll put that video and the presentation for the topic number 1, development of pickle from yellowfin tuna, right? And the changes in the nutritional quality.
Madam, am I audible?
Okay, Afrin. Okay.
Thank you.
Okay.
Good afternoon, all of you. I'm Afrin. I'm very glad to be here as one of the presenter in ISA 2023 symposium. Today, I would like to present on development of pickle from yellowfin tuna, mussel, and changes of nutritional content during the storage period. So my contents are introduction, objectives, materials and methodology, results and discussion, conclusion, references. Let's move to the introduction. Yellowfin tuna is a large pelagic marine fish in the Scombridae family. As fish is naturally perishable, it is essential to store it after harvesting. Developing countries are finding it increasingly difficult to protect fish and other marine harvest during large capture periods in order to satisfy the consumer requirement when the grab is insufficient. In recent years, there is a growing demand for the value-added fishery products due to the increased urbanization, social and cultural changes. Pickling is a safe, lowest, and easy method of putting up fish for short-term storage. So pickling is defined as the process of soaking food in a solution containing salt, acid, or alcohol. It would serve as a source of raw material for fish pickle that may provide a good taste and nutrition to the country and outgoing people at cheaper price. Pickle has a long shelf life and a minimal risk of food poisoning. The objective of the study to create pickle utilizing yellowfin tuna and decide the shelf life of pickle after incorporating various levels of vinegar at ambient temperature. Let's move to the materials and methodology. First of all, the experimental design. Here we can see there are 4 treatments. So all the ingredients except the vinegar were constant amount, but the vinegar for the treatment 1, 10 milliliter, and 30 for the treatment 2, and 50 for the treatment 3, and 70 for treatment 4. The procedure, the fish 2.5 kilogram weighted fish were collected and it was gutted and processed. Then they were cut into small pieces. After that, we washed the fish fleshes. The large pieces were cooked and deboned. The fleshes were marinated without the bones for 30 minutes. And for the marinating purpose, we have added the turmeric powder, chili powder, and salt. Then we fried for 15 minutes. Afterwards, the garlic and onion paste were fried in the oil until it became the golden brownish color. Then cloves, cumin, salt, sugar, and vinegar were mixed with that fried mixture. The pre-fried fish tissue was heated until the vinegar absorbed. Then the mixed with tamarind extract. Then we cooled the mixture and stored in the sterile glass bottle. the proximate and chemical evaluation. For that purpose, we have evaluated the moisture content, ash content, pH content, and titratable acidity. For the organoleptic evaluation, we have evaluated the color, aroma, texture, appearance, sourness, saltiness, and overall acceptability. Statistical analysis. For the experimental design, the complete random design was used and ANOVA was used using the SPSS. For the mean comparison, DMRT was used, and for the sensory evaluation, Turkey test was used. So let's move to the results and discussion. First of all, the fresh fish biochemical composition. The parameters for the moisture, the value we got 67.8%, and ash content 2.3%. The pH value was 5.3, and titratable acidity 5.4. The histamine level was 10.6 milligram per kilogram. Next, moisture content. For the moisture content, we can see among the 4 treatments, the T4 got the highest value for the moisture content, 65.53. Next to that, the T3 got the highest. Here we could see from the fresh fish flesh, the moisture content was decreased because 67.8 that was decreased to 65.53. But along the storage period, the moisture content was reduced. Next, ash content. Among the 4 treatments, the ash content also highest in T4 treatment, next to that T3. But the ash content was increased along the storage period up to 8.8. The pH. So here we could see among the 4 treatments, the T1 got the highest pH, 3.55. But the pH also that reduced along the storage period. The titratable acidity. Among the 4 treatments, the titratable acidity was highest in T4 treatment, 6.12. That was increased during the storage period up to 7.05. Next, sensory analysis. Among the 4 treatments, we could see the color, appearance, texture. The 3 variable got highest in T3. Next to that, T2 got the highest. But the value for the every variable were decreased during the storage period up to 15 days. Then aroma, sourness, and saltiness, all got the highest value for the treatment 3. That means vinegar 50 milliliter. But overall acceptability also high in T3, 6.8. That also reduced up to 6.6. So up to that, according to our study, we could conclude that the nutritional composition of yellowfin tuna fish pickle did not deteriorate during the storage at room temperature. It can be concluded that the choosing a procedure that links fish cooking and the addition of salt, tamarind, spices, and vinegar to low moisture pre-cooked fish chunks allows for the preparation of yellowfin tuna fish pickle with considerable sensory value and a better shelf life of 15 days at room temperature. The T3 was the best combination according to the sensory analysis. Yellowfin tuna fish pickle has the possibility to become an indigenous fish product that is ready to eat and extremely acceptable. So they are the reference which we have used for our study. And thank you so much.
Okay, thank you, Afrin, right, for your presentation on fish pickles. Any questions from the audience or the participants? Our panel member, Dr. Champati Gunatilaka, any comments or anything you want to say here? on this presentation? Afrin, please minimize the slides.
Okay.
Okay, Afrin, you have 4 treatments with different vinegar concentration, and you said 50 milliliter. For what is the weight of the fish?
2.5 kilogram.
For 50 milliliter.
No, divided by 4.
What divided by fish weight or?
No, fish plus or each treatment you are asking.
No, I am asking the 50 milliliter vinegar for how many? What is the weight of the fish?
All are equal amount weight.
What is the gram or kilogram? What is the
Gram.
weight? What is the weight you have taken?
Around 600 gram.
600 grams.
Yeah.
Okay. Then you added 50 milliliter.
Yeah.
Right, but when you compare the nutritional and the sensory, the ash content and other parameters are high in T4.
Yeah.
Yeah, but based on the sensory, you say that T3 is the best treatment, and also the pickles we can preserve for months and years. Sometimes we could, we can preserve for more than a year. But anyway, why your studies only you have conducted for 15 days, no? You can try and extend the period of storage, and then you try, and definitely I'm sure that you can keep the product more than 2 weeks. Okay.
Okay.
Any questions? Yeah, one more question.
Hello, Afrin. I have few questions on your methodology. I have seen that you use different volumes of vinegar as a treatment, isn't it?
Yeah.
So why didn't you go for concentration? Go for different concentrations, or what is the concentration of the acid used?
One
N. I don't get you well. Is it 1
N?
molar, 1 m?
1 N.
So you use same concentration, but with
same
different
concentration.
volumes.
Yeah.
Okay, and the other thing is, so you evaluated the texture and color under the sensory evaluation. So basically, I don't know whether I'm right. So the texture can be analyzed by the
Texturometer?
food texture analyzer.
Oh, texturometer?
Yeah. So why didn't you have the facility to use such instrument?
No, we haven't any that facility that time.
Or maybe you measure the hardness, no? Because the texture
the
analysis,
penetration.
hardness, you give different scales, from 1 to 5 or 1 to 7, like the overall acceptability, the hedonic scale we are using now. So likewise, the texture scale you give different, and then you can measure the texture and the color also, colorimeter.
Yeah, we have in our department, we have the color meter. We should have used it. I don't know whether
this
you know
was
that.
During this period we didn't have that
Such instruments.
Instruments.
Okay, so with limited resources you carried out this research. Okay, that's a good thing you did. But as Professor Deva Himayendran told, so if it is a pickle, it has to be stored at least for 1 year. But your study shows that it can be stored just for a very short period, just in 2 weeks' time only. It has a shelf life. But you should think of extending this research for keeping it for longer periods by trying some other preservatives or some other things. Okay, then that's a very good initiative work. So, Madam, you can
Thank you, Afrin. And you can,
Thank you,
try
ma'am.
for 1 year, and then you measure the quality parameters and the sensory qualities once a month and see how long you can preserve the fish pickers. Okay, with that, any other questions from the audience or the presenters? Thank you, Efren.
Thank you, ma'am.
Thank you.
Thank you.
Okay, one more, the last. WAMK Adhikaram, are you with us?
Yes, not joining.
I think the presenter has not joined. Okay, we play the video. The title is the determinant and impact of new technology adoption for paper post-harvest processing of farmers' well-being, a case of paper farmers in the K-Golf district. Okay, this is a research carried out at the Sabragama University. I think the presenter has only sent the slides, right? Anyway, we'll move the slides and so this is the second slide. Okay, that's only the slides the presenter has sent, and very sorry about that. He has not sent the recording. So we had 4 presentations. What is it? Okay. Okay, we had 4 presentations in the session on food nutrition, preservation, value addition, and post-harvest technology. So 3 of them had presented in the as person in the research finding, and 1 we have played the recording. And thank you for all of you, right, for your valuable informative research findings that you share with us in the International Congress ISA 2023 of the Eastern University. And we will upload the proceedings, right, very soon, in a day or 2 in our website. Or any queries, you can communicate with our coordinator, Mr. Rajendran. And thank you for the panel members. Dr. Champati Gunatilaka and Dr. Bavadarani. Dr. Champati, do you want to say anything to the presenters or the audience?
Yes.
Yeah, sure.
Okay. May I give my comments about presentation? Is it okay now?
Yeah, sure.
Oh, okay. Hope you can hear me well. Actually,
Yeah.
that I found the presenters did some good work under their research projects. So that the final presentation, actually, that is I observed that. But anyhow, that presenter required to be at least Q&A sessions need to be there. So then we can verify that what is the presentation. Otherwise, in overall, it will be good. Actually, so many different type of research findings it is presented in this symposium. That will be great opportunity for that presenters as well as other stakeholders. This kind of symposium. Okay, thank you very much.
Thank you, Champati, for joining with us for the session on food and nutrition. And other panel member, Dr. Bavadarani. Yeah.
First of all, I would like to congratulate all the presenters for presenting their findings at our symposium, ISA 2023. And it's very enjoyable and very informative, and we got different message from your findings. And one thing I would like to let you all know is when you submit paper, so it should be presented at least somehow. If the person cannot make it, he or she can arrange someone else to present at least. And that is a professional way of doing things. So I actually appreciate all the 3 presenters here. They came forward and presented their findings in a nice way. And good luck and congratulations.
Okay, thank you, Dr. Mrs. Bavadarani, the panel member of the session on food and nutrition. So thank you for all joining and for presenting your research finding. So we will conclude the session, technical session 8, and we will join for the section 2 on the same session very shortly. Okay, thank you for the participants and thank you for the presenters. Thank you. Have a good day. Enjoy. Thank you.
Thank you all.
Thank you.
Okay, thank you.
Close.
Recording stopped.